The topic for the present video is handling a new nitrosamine impurity in pharmaceutical products. There are many videos available on this channel for nitrosamine impurities, and this video is regarding the new nitrosamine impurity. If you encounter such a problem, or if you come across a situation where you get the new nitrosamine impurity, or you found the new type of nitrosamine impurity, or you found that the nitrosamine impurity is generated in the formulation. So how to handle this situation? This video will guide you for the step-by-step procedure to handle the new nitrosamine impurity. So let's start with the video. See, handling a new nitrosamine impurity is critical, as nitrosamine impurities have very stringent limits. It is a very critical task that involves many steps to ensure the patient safety, regulatory compliance, and maintaining the product quality. So, patient safety, regulatory compliance, then communication about this new nitrosamine to the regulatory authorities and also to the different stakeholders is very difficult, and it is very important. Considering the new nitrosamine, maintaining the product quality becomes very much critical. This video will give you the step-by-step approach to manage the identification and control of new nitrosamine impurity in the formulation. Many steps are there. Some steps are very elaborative, but to make the long story short, I have enlisted some of the points, and I assure you that after watching this video, you will get complete clarity about this topic. Handling a new nitrosamine impurity involves steps like identification and risk assessment, analytical method development, establishing control strategies, regulatory reporting or regulatory communication, you can say, then toxicological assessment, product reformulation and process change if required, quality control and batch release, risk communication, continuous monitoring and improvement, and the product recall or recall management if it is required. So many steps are there for the handling of new nitrosamine impurity. Out of that, the first step is identification and risk assessment. It involves root cause analysis, risk assessment, cross contamination evaluation. Root cause analysis involves the steps to identify the source of the nitrosamine impurity. It could be formed during API synthesis from raw materials, solvents, reagents, excipients, or due to the storage conditions during manufacturing process. So many reasons may be there. Coming to the risk assessment, conduct a risk assessment to evaluate the potential for nitrosamine formation. If you found the root cause, then a risk assessment is to be done. This may include reviewing the manufacturing process, degradation pathways, and storage conditions. Coming to the cross-contamination evaluation, assess the potential for cross-contamination during manufacturing and storage, especially if the facility handles multiple products. This means that if the product is known to be a non-risk type of product, but still you found new nitrosamine, then there may be the chance of cross-contamination. So whenever the product is being manufactured in the facility where other products are also handled, so cross-contamination evaluation becomes necessary. Analytical method development involves analytical method development, validation, checking its sensitivity and compliance to the ICH M7R2. Analytical method validation involves develop and validate sensitive analytical methods such as gas chromatography, GCMS, LCMS, or mass spectroscopy, or other suitable techniques for accurate detection and quantification of the specific nitrosamine impurity. Then sensitivity. Ensure the method is sensitive enough to detect the nitrosamine impurity at very low levels, typically in part per billion. See, the sensitivity is required to be very much very much uh, important, because these nitrosamines are potent and they have very stringent limits. ICH M7 compliance. For this, follow the guidance provided by ICH M7R2 for mutagenic impurities, which includes control strategy and analytical method for impurities like nitrosamines. So ICH M7 guideline is there to guide you regarding the analytical method. Then coming to the establishment of control strategies. Control strategy involves specification limits, process optimization, and control points. Specification limits. So set acceptable intake, that is AI limits for the nitrosamine impurity based on toxicological data in accordance with guidelines like ICH, M7R2 and regulatory bodies guideline EMA, FDA. All the guidelines are required to be taken into consideration for setting the specification limits. Process optimization. Modify manufacturing process to reduce or eliminate the impurity formation or impurity generation, and this could involve change in the reagents, solvents, conditions for raw materials and also for the APIs. Control points. Implement control points during manufacturing to monitor the levels of precursors that could form nitrosamines, ensuring that they remain below the acceptable levels. Regulatory reporting involves regulatory notifications, risk mitigation plan, and communication with health authorities. Regulatory notifications involves report the detection of new nitrosamine impurity to relevant regulatory agencies. For this, consider the product market and then know your regulatory agencies like FDA, EMA, or country specific regulatory body, and check their guidelines and make it in line with the regulatory guidelines of the specific country. Risk mitigation plan. Submit a detailed risk mitigation plan or risk assessment report, including analytical data, potential sources, and measures taken to control the impurity. So whatever the study you have performed during the previous stages, all those you have to enlist, report it, make a report, and then submit to the regulatory authority. Communication with health authorities. Engage with regulatory bodies for guidance on acceptable limits and corrective actions if needed. Agencies may require risk assessment or additional testing data. Regulatory bodies may have good knowledge about the specific nitrosamines, because many of the applicants are in continuous communication with these health authorities. So these health authorities can help the applicant. So communication with the health authorities about the new nitrosamine is very important. Toxicological assessment of the impurity. It involves carcinogenicity evaluation, acceptable intake calculation, and read across approach. So carcinogenicity evaluation means perform a toxicological assessment to determine the potential carcinogenic risk of the identified nitrosamine impurity. This often involves data from scientific literature or studies. So this carcinogenicity evaluation should be done, and then you can say whether the nitrosamine impurity is having carcinogenic nature or not. Then coming to the acceptable intake calculation, this is based on to the toxicological data. So based on the toxicological data, determine the AI limit or acceptable daily intake limit or permissible exposure limit for the nitrosamine impurity. Read across approach. So for this, I have made a separate video. You can watch that video and get the good idea about this read across approach. So if specific toxicological data is not available for the nitrosamine impurity, you can use read across approach from structurally related nitrosamine to establish the safe limits. You can also have the interim AI limits. Then product reformulation and process changes. So if at all it is not possible to control the nitrosamine limit, then reformulation is required. Formulation adjustment and manufacturing process changes are the stages for product reformulation and process changes. For formulation adjustment, consider reformulating the product to replace or remove materials that may contribute to the formation of nitrosamines. Regulatory authorities also have given some points to control the nitrosamine formation. Manufacturing process change may involve modification of the manufacturing process parameters to avoid conditions conducive to nitrosamine formation, such as pH adjustment, controlling temperature, and reducing the residual solvents. Quality control and batch release. So batch testing is there, release criteria is there, and stability testing is there. Implement the routine testing for the finished product and if necessary, intermediate steps for nitrosamine impurities. Release criteria. Ensure that the nitrosamine impurity levels in each batch are below the established safety limits before release. Stability testing. It involves conducting the stability studies to monitor the nitrosamine levels throughout the product shelf life so that you can ensure that no significant increase is there in the nitrosamine impurities during this shelf life period or storage period. Coming to the risk communication, internal training and external communication. These are the two steps for risk communication. Internal training means train the manufacturing and quality assurance teams and different stakeholders on handling nitrosamine impurities, including the risk and preventive measures. So training should be there. Then external communication. Inform the stakeholders, including healthcare professionals, about the impurity and the steps taken to mitigate the risk and any implications for patient safety. So this is the external communication. Continuous monitoring and improvement. There should always be a continuous monitoring and improvement for the well-being of patients and for controlling the nitrosamine limits. Post-implementation review is there and staying updated with the guidelines. These are the two main points for continuous monitoring and improvement. So post-implementation review involves regulatory review for the effectiveness of control strategy for nitrosamine impurities through internal audits and continuous improvements. Staying updated with the guidelines. So keep up to date with the evolving regulatory guidelines and scientific literature related to nitrosamine impurities and adjust control strategies accordingly. These are the strategies. Recall management. So if necessary, the recall management system should be there in place. A risk to the patient. If the levels of the nitrosamine impurities exceed the acceptable intake limits and pose a risk to patients, implement a product recall in accordance with the regulatory agencies. This is very important point that the manufacturer or supplier of the medicinal product should have a recall management system so that the public health can be secured. Corrective actions. Implement corrective actions before resuming production to prevent a recurrence of the impurity issue. This is very, very important and very critical aspect. What the kappa you have taken? You have done all the steps, but what is the kappa? So corrective action should be there in place. See if the new nitrosamine is generated or found in the formulation. It is a very big thing. It is very critical. So effectively managing nitrosamine impurities requires a proactive approach involving detailed risk assessment, methodological control strategies, and regulatory collaboration to ensure the safety and efficacy of the pharmaceutical products. And this is critical because nitrosamine are mutagenic in nature. These are potent impurities These are carcinogenic. That's why nitrosamine impurities are very much critical. Handling the new nitrosamine is very important and very critical for the patient safety. This video is regarding the necessary steps. If you get knowledge about the other steps also, please do comment so that I will also learn about this topic. See, as a pharma professional, I am also learning these things, and whatever learning I get, I wanted to share with my pharma colleagues. So, if you found anything regarding the steps for new nitrosamine handling, then please feel free to comment so that from those comments everyone will get benefited. I hope I have covered the topic in detail and you might have got good idea for the steps for handling nitrosamine impurity, specifically the new nitrosamine impurity. And in upcoming videos, I will elaborate these steps in detail. Thank you for watching the video and thank you for being with the channel.